Hello, this is Mrs. Kohler. Now, we've been talking about the cell cycle, but not very much detail. So today what we're going to do is we're actually going to describe exactly what it is that's going on during the cell cycle. Because you're going to have to know the steps, and then you're also going to need to be able to do this with a set of pop beads for lab. So before we can talk about the cell cycle, we need to talk about a couple of basic structures that you need to know. And these are on your note sheet, so you can just fill this in as you go through the note sheet. So this is what the cell looks like before it starts to divide. So you have the nucleus. The nuclear membrane is that barrier around the outside of the nucleus. The nucleolus, remember, is like a mini nucleus inside the nucleus. It's a darker area inside. And now you have a new structure called chromatin. So about chromatin. It's a mass of DNA and proteins, and those proteins have a special name, which are histones. And it's the form that DNA is normally in. DNA only forms up into chromosomes for cell division. The rest of the time it's unwound. And this is an electron microscope image of chromatin. So the reason that it has to be unwound most of the time is that in order to copy the genes for the cell to do what it needs to do, it has to be unwound. It'd be like trying to play with a toy that you get for Christmas before you take it out of the box. You can't do that. You need to take it out of the box, and that's what unwinding it into chromatin is like. Now, when DNA does condense into a chromosome, you have a couple of different structures in it. So the chromosome itself is just compact DNA structure. So you take the DNA that starts out as just strings, and then it starts to wind up into little curlicues, and then those curlicues get more and more condensed until finally you end up with a structure that looks like a great big X. That's a chromosome. So the chromosome consists of two halves, and each half is a sister chromatid, and they're called sisters because they're identical to each other. So if you look here on the blue side, this would be your original, and then during the S phase it makes a copy of itself, which would be the yellow side. So they're identical, and each side occupies one half of the chromosome. They're sister chromatids. And they're joined in this at the centromere, which is this central area right here where they're held together. So you're going to need to know those parts of a chromosome. Now, we're going to talk about the cell cycle and specifically what mitosis does within the cell cycle. So here's your cell cycle diagram again. You don't have to copy this. You already have a simplified version of it in your notes. So we're going to start with cell growth, which is interphase. So the period of cell growth is interphase. This is where the cell spends most of its time, and it has three parts, G1, S, and G2. And for this part, you're going to want to make sure you write small and abbreviate, paraphrase a little bit. So the first stage of interphase is G1, also called gap 1 or growth 1. So this is where the cell does what the cell does. Proteins are made, genes get expressed, the cell grows, and it does whatever that cell's supposed to do. During S phase, remember S stands for DNA synthesis. So during S phase, the cell replicates its DNA, or it makes another copy of its DNA. And the reason it does that is because in order to divide, each new cell is going to need a full set of instructions. So it needs to duplicate in S phase so that each cell has a full set of instructions after it divides. And then G2, which is also gap 2 or growth 2, the cell grows some more, and also the organelles duplicate. So you're going to make more mitochondria, you're going to make more ribosomes, you're going to make more chloroplasts if you're a plant. This is preparing for division. So this is what the cell looks like in interphase. You basically have a cell, a normal looking cell with a nucleus. Here's some other versions of it. Now this one up here is an animal cell. So you can see the nucleus right here really clearly. Whenever you can see clear nucleus, it's probably interphase. And this one here is a plant cell, and you can tell because of the boxy cell walls, and the real clear nucleus lets you know that it's an interphase. Now, so that was interphase. Now we're going to talk about the 4 stages of mitosis. That's the division of the nucleus. And it's going to result in 2 identical nuclei. Not 2 new cells, just 2 new nuclei. So the 4 steps are prophase, metaphase, anaphase, telophase. And remember the acronym PMAT to help you with that. So during prophase, this is what the cell's going to look like. The nucleus prepares to divide during prophase. So the DNA is going to turn from chromatin into chromosomes, so it's going to condense. The outside of the nucleus is going to disappear. The centrioles, which are these things here, they're going to move to either side of the cell. And the reason they're going to do that is they're going to produce the spindle that's going to guide everything to where it needs to be. And spindle fibers, which are these kind of stringy things coming out from the centrioles, start being produced. And they're actually going to join to the center of each chromosome in order to guide it to where it needs to be. So this is an animal cell in prophase, and you can kind of start to see the nucleus is breaking down and you're starting to see little X's in there. And same thing with the plant cell over here. You're starting to see like little holes in the nucleus, and that's because the DNA is condensing. So when you start seeing holes in the nucleus, you know that it's prophase. Now, PMAT. We did P, now we're going to do M, which is metaphase. And when you think metaphase, think M for middle, because what's happening is that the chromosomes are actually lining up down the middle of the cell. So the chromosomes line up at the middle of the cell, so it starts looking like a row of X's, and each centromere gets attached to a spindle fiber. So up here you can see these strings are going to the middle of the chromosome. That's the centromere, and this is the spindle fiber. Now in the animal cell, this is a really great picture. It almost looks like a black line straight down the middle, and that's metaphase. Now in this one, in the plant cell, they're not quite completely organized yet. They're starting to get there, but some of them haven't quite gotten there. But you're starting to see X's, and they're starting to get to the middle. So that's going to be metaphase. Anaphase is the next one, PMAT. We did P, we did M, now we're doing A. When you think anaphase, think A for apart, because the chromosomes are going to pull apart from each other. So here's where they were for metaphase, and now they're pulling away. The sister chromatids separate from each other so that one goes this way and one goes that way. And they move away from each other towards the poles of the cell. And think like the North Pole and the South Pole. This is one pole. This is another pole. So they're going to move towards the centrioles to opposite ends of the cell. So in the animal cell, you can see real clearly that you have a group over here and a group over here, but they're not a nucleus yet. So that's anaphase because they're pulling apart. And for the plant cell over here, you can definitely see there's a division in the middle here. They're pulling apart. Now, telophase. Telophase, think like T for 2. When you talk on the telephone, there's usually 2 of you talking. So it's the last step in mitosis, and there's going to be 2 nuclei, 2 nucleuses, which is nuclei. So the centrioles and spindle fibers are going to go away. The chromosomes are going to start relaxing again, and at the end of each cell, you start to get an actual nucleus forming. So you have one cell, but there's a nucleus at each end of it. So T for telophase, T for 2, you have 2 nuclei. So you can see here in the plant cell, you have a nucleus at each end. You have a little bit of a cell wall starting, but that's really cytokinesis. But the 2 nuclei that look like they're still in one cell, and here you have a little bit of cleavage, but the cells aren't completely done forming, so you're still a little bit in telophase here. So, quick practice, finding the different phases. Well, let's see. Interphase, what we want to be looking for something that has a nucleus and a nucleolus. So here you have this. Here you have the nucleus, and you have the nucleolus real clear in it. So that's going to be interphase. Now, prophase, you can start seeing little holes in the nucleus. Prophase is starting. You see a little bit of areas that are a little bit more condensed. That's early prophase. Metaphase, they start lining up in the middle, so this would be metaphase down here. because you're starting to see the X's and they're starting to go towards the middle of the cell. Anaphase apart, this is usually the easiest one to find, and see how you have the distinct division between the two. And then telophase, you have what looks like one big cell and two nuclei inside of it. That's telophase, so T for 2. Now, after telophase, and actually this sort of starts during telophase, not quite at the end of it, but there's a difference between telophase and cytokinesis. Telophase is when you have two full nuclei. Cytokinesis is just when the cell itself divides. So you already have the two nuclei. You have one here, and you have one here. So you already have the two nuclei. But now the actual cell is dividing. The cytoplasm is dividing. So that's cytokinesis. Now there is a difference between cytokinesis in plant and animal cells. In animal cells, you get what's called a cleavage furrow. And I know all the gentlemen will know exactly what I mean when I say cleavage, because that's what it looks like. It looks like two circles being pinched in with many circles inside of them. It's a cleavage furrow. It starts pinching in. Plant cells can't change their shape so easily, so they get what's called a cell plate. And you actually get a new cell wall growing without any of that pinching in. So it really stays very square in plant cells. So plant cells get a cell plate, animal cells get a cleavage furrow. And that's what the cell looks like in cytokinesis.